In this lecture, we're going to keep discussing about the protein, but especially focusing on the protein structures in more details, and also we'll discuss components to regulate such structure. Let's start with a summary of levels of a protein structure. So here's a peptide bonds that links together of the amino acid to form the primary structure of the polypeptide. And also, there can be hydrogen bonding interactions within the backbones of those polypeptides to form the secondary structure. And within this secondary structure, the side chains of amino acids within this polypeptide can interact with each other to form this 3D structure, which is a tertiary structure. And finally, multiple polypeptides can get together to form the quaternary structure, also by utilizing the side chains of the amino acids within this polypeptides with the other polypeptides. And this folded protein structure is stabilized by a variety of weak chemical interactions, such as electrostatic interactions, as well as hydrogen bonds and the van der Waals forces. And in some cases, those interactions can be stabilized by the covalent bonds, especially the disulfide bond between the cysteine residues, that we're going to talk much more in details about their role and also their mechanisms to form the tertiary structure as well as the quaternary structure. Here's another summary of the protein structure with a more detailed overview here in the table. So there are mainly 4 different types of protein structures, and this table particularly includes 2 more, which is super secondary structure as well as domain here. So first, take a look at the primary structure, which is basically amino acid sequence of the protein that forms the polypeptide. And the secondary structures are the alpha helices, beta sheets, and the turns and loops that the particular shape that part of or the whole of the polypeptide makes. And super secondary structure is basically the mixture of these secondary structures. So association of secondary structures such as alpha helices and beta sheets together. And the domain is a part of a polypeptide that can be worked as a module, actually a functional group. So it's a self-contained structural unit and typically have a particular function and gives a special feature of that protein or the polypeptide that contains that such domain. And tertiary structure is again the folded structure of the whole protein or the whole polypeptide, and it also includes the intramolecular disulfide bonds that was formed by the side chains of the amino acids within that polypeptide. And lastly, the quaternary structure is assembled complex consists of multiple polypeptides. So it can be also called as oligomers. And there are 2 different types of oligomers. The first one's a homo-oligomeric structure, which consists of just one protein type. And also there's a hetero-oligomeric structure that comprises with more than one type of protein as a unit for the NDM that works as a building block. The polypeptide bond that is formed between the carbon and the nitrogen of the 2 different amino acids have a planar character, meaning that they tend to stay flat, and that is due to the partial double bond that is formed between the carbon and nitrogen atoms, which prevents the peptide bond from rotating too much. And that is because of this double bond that is formed between the oxygen and the carbon that resonances with this carbon-nitrogen bonding too. So in consequence, they're going to form this pi orbital up and down of the plane where the carbon and nitrogen forms the bond. So they're going to have these specific angles and try to maintain that angle because of these electron densities up and down of the plane. As a consequence, when you look at the multiple protein sequences within the polypeptides, they tend to align like this to form the specific angles between different amino acids to minimize the steric liaisons. So the regions where the peptide bonds are formed are pretty rigid due to the alpha orbitals located up and bottom of the planes. But polypeptide or the protein can still have a flexibility due to the rotation around this alpha carbon. And again, alpha carbon is the center of the amino acid where the side chain or the side group is attached. And depending on how these two bonds, which is to the nitrogen and the other carbon, is twisted or the located or the rotated, that can define and also assume the different secondary structures such as alpha helix and beta sheet, which we are going to discuss more in a few slides. So the conformation of the backbone itself here can be simply described by these two values, phi and psi angles. But there can be a steric clash, especially when this oxygen and the nitrogen atoms are on the same side, where they're going to be a reference point for the psi and phi values, as you can see here. And this is improbable. This is impossible structure. So there are not going to be any cases where both phi and psi values are zeros. Ramachandran plot shows the probability of the secondary structure due to the phi and psi values on around the alpha carbon on the polypeptide. So as you can see, the phi value is usually negative, and it's mostly likely formed the beta sheet or the right-handed alpha helix. And there's cases where the polypeptide makes the left-handed alpha helix too, where the phi value is rarely the positive values and the psi value is positive too. And again, this is where the phi and psi values are located and described. And this phi and psi value indicates the rotation again around the alpha carbon for each amino acid residues in the polypeptide, meaning that whenever you have alpha carbon, you're gonna have a phi and psi value for those residues too. So by having all the information about these phi and psi values, you can actually simply build this polypeptide into the secondary structure pretty easily, and the 3D structure too. And this rotation allows the polypeptide to assume its various conformations such as beta sheet, alpha helix, as well as just the loose loops. And some conformations of the polypeptide backbone results in steric clashes. An example was that both phi and psi values were zero, which is improbable right here. Between the atoms, the oxygen and the nitrogen atoms that I made as an example in the previous slide. And this is simply disallowed. And glycine, which you can recall its structure, the smallest one with no side chain or just a hydrogen atom, is therefore have the conformationally highly flexible form there, so it can make the loops, or can be involved in the beta sheet or the alpha helix too, but it's pretty free, so it can form any loose structure. The first protein secondary structure that we're going to discuss more is alpha helix, and it can be shown as a ribbon here, which indicates the backbone of the polypeptide. And this is particularly the right-handed alpha helix. And you can see the carbon, alpha carbon in the main chain too. And also amide planes, which indicates the peptide bonds. And also there's a hydrogen bonding that are formed between these backbones that spans the alpha helix to stabilize this secondary structure. And usually there are 4 amino acids that are involved within one turn of the alpha helix. As you can see here, alpha carbon I will be on the same position as alpha carbon I plus 4. And all the side chains, mostly the side chains, which are R groups of the amino acids, point outwards of the alpha helix to minimize any kinds of steric clashes. So when you go back to the Ramachandran plot, you can actually see that again, the phi and psi values are both negative, and around 50 to 60 degrees or so. And in addition to hydrogen bonding between the atoms of the backbone, the stability of the alpha helix is influenced by the side chains, the R groups interactions such as charge-charge, van der Waals, hydrophobic interactions, as well as steric interactions too. And small residues such as alanine, but not the glycine because it's the littlest one, and it's mostly involved in the loops. The alanine are more common than the bulky residues, which can actually make some steric clashes within this kind of tight, cramped structure. There are several different ways to show this secondary structure, the alpha helix of the polypeptide. One is using the balls and sticks, as you can see. It also indicates where the hydrogen bonding happens, different types of atoms that are located, as well as the side chains too. And you can also show that with the ribbons, but with the this backbone indicated or without it too. And also there can be a 3D structure of the protein too. And another way to present and visualize the alpha helix, especially the position of amino acids around the alpha helix, is the helical wheel, as you can see here, in which case you can see the components of amino acids, components of the alpha helix, each amino acids, where they are located and so on. And it also allows for a quick visualization of whether a side of an alpha helix possesses specific chemical properties, such as hydrophobic properties or the basic properties. And the example shown here is the alpha helix that forms a leucine zipper, which has a critical function for interacting with the DNA molecules. And when you look at it closely with the helical wheel for that leucine zipper alpha helix, you can actually see the hydrophobic residues such as leucine is on one side to interact with another helix right here that displays the same pattern. So leucines will be on the one side. So those two hydrophobic residues can interact with each other to make something like a zipper, have a strong interaction with them to hold them together. Most amino acids like to be in an alpha helix because they have hydrogen atoms as well as oxygen atoms within their backbone, so it tends to form the hydrogen bonding, as well as the side chains can interact with each other due to the some other weak interactions to form this alpha helix. But there's a notable exceptions. The first one's glycine, the amino acids with the smallest side chain, which is just a hydrogen atom, and it can actually generate the wide array of phi and psi values, meaning that it can be so too much flexible, so it makes the alpha helix hard to sustain its form. And proline is another exception where it has a special structure, as you can remember. So this proline is pretty rigid, and also there's no hydrogen atom attached to this nitrogen, so it's not going to form the hydrogen bonding with the other side chains or the backbone. Therefore, the proline residues often serve as alpha helix breakers because it cannot form the alpha helix and usually just disrupts that structure. And both residues, the glycine and proline, are often found at the boundaries of the alpha helices and in turns too, working as a breaker. The alpha helices are the major structural components that many proteins contain, and also these alpha helices are connected by the turns usually. So one protein can contain multiple alpha helices. And an example is in the hemoglobin, 70% of the residues actually form the alpha helices as their secondary structure. And these alpha helices have a DNA binding functions. So an alpha helix fits perfectly into the major groove of a double-stranded DNA, but not the minor groove, but the major groove, it just fits right in there. So you can interact with a lot of DNA, the nucleotides within the nucleic acids there, using the hydrogen bonding as well as some other weak interactions too. And the leucine zipper, it can be one example here too. And in many cases, there are different residues within the helices are designed in a way that it can recognize a certain specific DNA sequences. So it is widely used for the DNA binding protein that has a specific target for a specific gene or the region within the chromosome. And also, there are the alpha helices that spans the membrane too, in which case the alpha helix contains the hydrophobic amino acids right in the center of this helix to allow the protein to cross the bilayer membrane and can be stuck there and stabilized by interacting with other, the hydrophobic interactions. And alpha helices can also be amphipathic too, which means that it has hydrophilicity as well as hydrophobicity at the same time. These helices possess hydrophilic amino acids or the residues on the one side and the hydrophobic residues on the other side. So depending on how this alpha helix is facing, it can be either hydrophilic or hydrophobic, and its interaction will be different. So these alpha helices, the polypeptide, can be especially stuck and snuggle into this lipid bilayer in a lateral way like that, with the hydrophobic regions that is facing to the lipid bilayer region. So it's interacting with the aliphatic carbon chains in this region. And as well as the other side, the hydrophilic side, is exposed to the aqueous solution or the condition within the cell. So it can be stabilized as it is for this location as well as structure too. And these helices, in some cases, can be used to associate a protein to a membrane. So there's a case that some other soluble protein that is hydrophilic needs to be on the near this lipid bilayer. It can actually use this amphipathic helices as an anchor, so it can be bound here first and can do its biological function to the other regions of the bilayer or the lipid bilayer itself too. A second protein secondary structure that is also very common is beta sheets. Unlike alpha helix, which forms a sort of the spring-like structure, beta sheet forms a pleated structure where all the amino acids within this polypeptide is aligned nicely on the relatively the same plane. And the basic unit of the beta sheet is called as a beta strand. So here, this one string of a polypeptide is one beta strand, and this is another beta strand. And of course, these two beta strands are connected to each other as a part of a huge polypeptide. And as you can see, the direction can be different here. So for example, the N-terminal region comes here. So naturally, the C-terminal of this particular amino acid will be located here. And then, since it is connected to each other, so the next amino acids will face in a way that the N-terminal of it will be facing on this downward and goes up there to get the C-terminal region on here. So they're going to be on different termini that you're going to see on the neighboring beta strand. It's called antiparallel beta sheet. And also you can get the parallel beta sheet where you can see all of them are aligned in a way that the same N-terminal regions are facing on the same end, and the same C-terminal also on the opposite regions. And if you go back to the Ramachandran plot, you can actually see that there are two different regions where the beta sheet can be formed based on this phi and psi angles, the degrees that you can see right here, but they are slightly different to form either antiparallel or parallel beta sheet. And these are the repeating unit, the beta strand, as well as the beta sheet itself, is the repeating unit of the just like on alpha helix. But the beta sheet can form various higher level structures or the super secondary structures where the different type of secondary structures are put together there to form the higher order like 3D structure, such as the silcofibroin and the beta barrels that I'm going to show you some of the pictures of that in a few slides. So here's again the top view of the antiparallel beta sheet versus the parallel beta sheet. As you can see, the antiparallel goes N to C, but the next one actually located as a C to N terminal, as opposed to parallel beta sheet shows the same direction, N to C, N to C terminal. Let's talk more about the beta sheets or the beta pleated sheets. The strands of amino acids are held together in sheets by inter-strand hydrogen bonding. So there are different beta strands that are aligned right next to each other to form the beta sheets. And between these strands, there are always hydrogen bonding that is especially formed on the backbone of the carboxylate group and the amino group in the different strands to held together to make that beta sheet structure stable. And strands of the beta sheets tend to be twisted a little bit and inclined, especially in the beta barrel, to form the higher order structures such as a supersecondary structure. And beta barrel structure is basically there's a multiple beta sheets that put together to form the sort of the outlay of the beta barrel, the barrel structure, to make the channel inside of it, and it is widely used structure for especially for the ion channels or some kind of transporters between the transmembrane. The beta sheets, but cannot be transmembrane or the transmembrane protein itself unless they're part of the beta barrel structure because of the little like natural polarity that build up on the backbone as an amino acid. So it has the polarity that inhibits from the incorporating into the membrane too easily. So it has to be sealed and hidden from these the transmembrane lipid bilayer. So to do so, it forms the beta barrel structure and expose some of the nonpolar regions that are facing to the membrane to make that structure stabilized. So the side chains or the R groups lie perpendicular to the sheets typically. So they basically stick out on either face of the sheet like this. And differential arrangement of R groups with the different properties such as basic or the acidic groups can produce a variable polarity. So it can actually work as an anchor for interacting with other proteins like soluble proteins or some kind of polar or nonpolar chemicals or the substances. So just like an alpha helix, beta sheet structure can be also described in different ways, several different ways here, using the ball and stick structure here, which can also indicate where the R groups or the side chain facing, as well as the hydrogen bonding and other atoms too. And also can be a hybrid form here where it uses the ribbon as well as some of the important ball and stick that indicates the backbone of the polypeptide, and also the 3D structure with all the molecules or the atoms indicated in this way with the color coded. Just like an alpha helix interacting with the DNA, especially on the major group, beta sheets also have an intriguing and interesting property for interacting with the DNA. So beta sheet fits very well, especially into the minor group of the DNA double helices. And you can imagine that as opposed to bulky alpha helix, that thin layer of the beta sheet can fit in really nicely into this little chasm, which is a minor group of the DNA. And beta sheet can also used in DNA binding, but are generally less commonly used. So alpha helix is the major mechanism where the protein and the DNA can interact with each other. But that doesn't mean that all the proteins require to have this alpha helix. Some proteins utilize this beta sheet to interact with it, especially with the backbone. Like we discussed earlier, there are two different types of beta sheet structures. One's antiparallel, and the others parallel beta sheet. And please note that particularly for the parallel beta sheet, you can see that the little linker here, or it can be also called as loops, are much longer than the one that is in the antiparallel beta sheet. Now we're going to turn to the a little higher order or the bigger scale on structure of the protein secondary structure, which is based on the super secondary structures, and they can form the higher order, again the bigger structure, which is typically a fibrous proteins, meaning that these multiple protein units put together to form the fibers, or sometimes it can look like a web kind of structure too. And they're basically elongated and filamentous proteins. So there are multiple proteins, the subunits get together there, and they play key and important structural role in animal tissues, basically serving as the scaffold for the other tissues to form into some kind of organs and stuff. And the examples include the skin and connective tissues and so on. And often they form it outside of the cell because they're quite bulky and work as a sort of adhesive for the other tissues too. And these are the three different examples that we are going to discuss more. And I'm just giving you this overview idea here, but we're going to go over these type of higher order structures one by one, and look at the more the details about their structures. So the first one is alpha keratin, and its monomer is alpha helix, and subunits of them form the strong, stretchy, and flexible intermediate filaments, of which hair, nails, and part of the skin are composed. And collagen is on the second example, and this is the most abundant vertebrate protein, and its monomer is an unusual left-hand helix. So remember that the most of alpha helix forms the right-handed, but this one's actually left-handed. But you're going to see more properties of this, how this left-handed helix contribute to the stability of this fibrous protein structure. Anyway, they are the remarkably strong and somewhat elastic fibers, and that forms the bone matrix, and their basic structure of tendons and important elements of skins too. And the last one is the silcofibrinoin, which subunit is beta sheet structure, unlike alpha keratin or the collagen. And they form the spider webs or the silk that is produced by the silkworms. So the first example for the fibrous protein is alpha keratin. This is just a one subunit of that, which forms the alpha helix, as you can see, where the N-terminal region is located on the top here. And you can also see it in from the top view, which is a helical wheel. And you can notice that this A, the first one, and the fourth one, D, as well as the other unit, are the highlighted in different colors. So individual molecules are long alpha helices, in which every fourth amino acid residues is hydrophobic. So these are hydrophobic. Thus, each helix has a stripe of hydrophobicity along one side. It's indicated in red here. So there's going to be one hydrophobic region exposed on one side of the alpha helix for one subunit, and the other subunit, which is the same alpha keratin, will also have this just one side of the hydrophobicity there. And using the hydrophobicity, pairs of these helices wind about each other in a left-handed fashion here. So unlike just a single subunit, which is right-handed, this dimer forms the left-handed helix now. So this is basically a coiled structure within this dimerization. They're stabilized by the interactions between the hydrophobic residues. And these dimers then associate to form the even higher order structures. So they're going to be a bundling of all these dimers too to make this structure really strong and rigid. So let's move on to the next slide to see that higher order structure. And here's the proposed structure for the alpha keratin intermediate filaments or higher order structure. Since this is a really bulky, really big structure, it was hard to actually validate that it actually forms in this shape. So there's only proposition for that for the structure. But anyway, its monomer of the filament is alpha keratin again. And how it assembles is that starting from the monomer, the single right-handed alpha helix, alpha keratin, but they make the dimers to form the left-handed coil-to-coil structure. And pairs of these coil-to-coils form the protofilaments, as you can see here, and that get also bundled to form the protofibrils. So they are the really flexible and very strong cable-like structures. And also alpha keratin contains large amount of cysteine, and the cysteine amino acid is particularly important for the biological functions. If you can recall that it forms the disulfide bond, the SS bond, within inside the molecule, as well as the between the different polypeptides too. So in this case, they form it between these protofilaments to stabilize in this structure for making the cross-linking in certain tissues. So it is rich in the nails, which is has the pretty rigid and the strong structure, but less prominent in hair. As you can feel, the hair is more smooth, so there's a less alpha keratin higher order structures formed there. And the second example of the fibrous protein structure is collagen. And this is a very interesting example because it uses the left-handed helix instead of right-handed helix, just like alpha keratin. So its basic subunit is a tropocollagen, as you can see here, just a one polypeptide, but a triple helix consisting of 3 left-handed helices. And there's a 3.3 residues per turn, so it's a little bit like more twisted. But they are coiled around each other in a right-handed direction. So the subunit or the monomer itself is left-handed, alpha helix, but they form the coil, 3 of them, on a right-handed manner. So, and they have a lot of glycine X, Y residues, where the X is usually proline, and Y is usually proline or the hydroxyproline. The repeat ensures the stability of helix. So when you look at the protein sequence here, they're pretty simple. They're mostly just a proline, proline, and so on. And subunits are packed together in a staggered fashion to form the collagen fibers that undergo extensive cross-linking too, using the disulfide bonds here between the cysteines in a different subunits or the units for this higher order structure. And the last example is the silk fibroin, also an interesting example for supersecondary structure because beta sheets are involved there instead of alpha helix. So they're composed of layers, multiple layers of beta sheets containing repeats of a sequence consisting almost entirely of glycine, alanine, and serine. And their interactions between them, especially between the layers of the beta sheets, facilitates the stacking. And layered beta sheets provide toughness and flexibility too. And another important protein or the polypeptide structure is turns or loops, which is pretty flexible and usually connecting two secondary structures such as alpha helix and alpha helix, or alpha helix and beta sheet, and so on. And there are various types of turns too, differing in number of residues and hydrogen bonding patterns too. And loops are typically longer, and they are often called as coiled. And most loops do not usually have a regular or repeating structures because they're pretty flexible and they don't want to actually form any kinds of rigid structures here, but only serve as a connecting other secondary structure. So they're quite like variable in terms of their amino acid constituents. But parallel strands in beta sheets are often joined by longer loops and that sometimes contain alpha helices too. So in that case, they're going to be a little bit more rigid and also quite regular in terms of their amino acid sequences too. And we're going to go more details about the different types of turns and loops. The first one that we'll discuss is beta turns, which is pretty typical, or you can see that pretty commonly in the protein, many of the biological protein structures. They are basically two different types for the beta turns, type 1 and type 2. And you should note the position, especially on the side chain of the second amino acid and the third amino acid, the R2 and R3, how they are located to distinguish between type 1 and type 2. So the type 1 turns have the R groups on the same side for the R2 and R3, as you can see here on the back side, as opposed to type 2 has these R groups in the opposite side. And residue 3 is usually glycine 2. And please note that the hydrogen bonding between backbones is formed on the residue first one and the last one. And another type of turn is gamma turns, which has 3 amino acid in its turn, utilizing proline in the middle at the turn. So it is a pretty rigid amino acid structure, as you know, as you can recall, the proline is, but it can actually provide a kink here. So only the 3 amino acid can still make the turn. And there's a hydrogen bonding between the residue 1 and residue 3 through the CO and NH group. And another example is omega loop, and it literally looks like an omega here, which involves more amino acids within the loop, so it's quite more flexible and quite bulky too. And this is an example of a protein structure of ribonuclease A, where you can see multiple alpha helices as well as beta sheets too. And between these two beta strands, there's a loop that links these two strands, and this is exposed on the surface of the protein for its functionality. So motifs are sort of the unit of the structure that can form by the one or more of the secondary structure of the polypeptides. So there are all different types of the motifs. Let's go over all these examples one by one. The first one is beta alpha beta, where the beta strands are connected by the alpha helix, and beta hairpin basically makes the pleated structure here with the multiple beta strands. And alpha alpha motif has the 2 alpha helices that interacting with each other, looking like some kind of zipper or the finger too. And there could be multiple beta strands that connected to each other, but instead of forming the beta hairpin, it can form into a way that it resembles the Greek key. And there's beta barrel motifs too. Like I told you, multiple beta strands can form this barrel-like structure, which leaves the sort of the hole in the middle or the channel. But it can utilize this anti-parallel beta strands or the beta sheet structure, or you can use the more like a Greek key structure too, and also it uses this alpha helix in between the beta strands to form the beta barrel structures too. And these multiple motifs or the supersecondary structures can form the domains, which is a sort of a modular unit within the protein or the polypeptide, can have its own function. So it has its own property there. And many globular proteins consist of several compact, locally folded, and stable regions called domains. Like I told you, this is a modular unit, so it's going to have its own independent functions in many cases. And domains tend to have about 40 amino acids, but it can go up to 350 too. But fewer than 50 amino acids will make it difficult to fold stably and stay stably, but more than 300 amino acids now make that polypeptide difficult to fold correctly too. So this 40 to 300-ish of the amino acid size is a sweet spot to keep it as a modular unit and have a specific functions pretty easily and keep it stable. A single domain is typically made of a single stretch of a primary sequence, but there are many exceptions too. So let's say that there's a one soluble proteins that can have the hydrophilic regions outside as opposed to inside of it. There could be some hydrophobic regions as a soluble protein within the cytosol. But for the protein that has multiple domains, now can have individual sort of a modular units there. Each of them can actually have a hydrophobic core as well as hydrophilic shells too. And again, these domains are modular, so this particular domain can be detached and attached to the other protein and can actually work pretty functionally because of this sort of independent structure. An example of the protein that have multiple domains is a glyceraldehyde 3-phosphate dehydrogenase, which structure is shown here, and it has two domains. One domain is interacting with NAD+, and the other is actually enzymatic domain that works as a phosphatase dehydrogenase. And we're going to discuss much more about this in the metabolism section. So far, we talked about the protein primary and secondary structures as well as super secondary structures, which is a mixture of the secondary structures. Now we're going to move on to the upper level in terms of a protein structure, which is a tertiary structure or the 3D structure of the polypeptide. In many cases, proteins are globular, especially the soluble proteins that are working, having the enzymatic actions in the cytoplasm. And they play a key roles in most chemical and many structural functions in the cell, and especially for chemical roles, they work as enzymes to process or accelerate some of the important biochemical reactions. And they possess compact tertiary structures generated by the folding and packing of secondary structure elements, super secondary structure elements, such as motifs and domains too. And tertiary folding is usually stabilized by a variety of non-covalent interactions such as hydrogen bonding, ionic interactions, van der Waals interactions, and hydrophobic interactions. Again, between the amino acids or the residues, but only the side chain, the R group interactions are at play, not the backbone of that. And this tertiary structure can be stabilized by the covalent bonds too using these disulfide bonds. So two cysteines are involved there. Using the oxidizing agent, it can form the disulfide bond and it's called a cysteine. Here are some examples of some globular proteins. And these are particularly viewing proteins as a secondary structure models where you can see the alpha helix, beta sheets, loops and turns, and so on. So the first one here is the helix bundles where the multiple alpha helices are stacked together, as you can see here, to form the bundles. And second one is the beta sandwich, which is similar to alpha helix bundles, but instead of the alpha helices, now using the beta sheets to stack them down to make this kind of compact structure. And third one is parallel beta barrel. It's also called the alpha beta sequence because of the alpha helix and beta sheet structure. And there's a twisted beta sheet here too, as you can see, a little twisted, which actually helps the stability of that protein structure too. And anti-parallel beta sheet structure is here too, and also combination of quite a lot of loops and the beta sheet and alpha helices and so on. And when you look at the side chains and it's their distribution, spatial distribution within the globular protein, you're going to see that most of the hydrophilic side chains located the outside of this globular protein, which are also going outward and interacting with the water molecules and some other hydrophilic reagents and the proteins too. As opposed to that, the hydrophobic side chains located more like the inside of the globular protein here, which is indicated in red and orange. So this table, which is table 6-3, shows the hydropathy scale for amino acid side chains. So it has been calculated of the hydropathy of the side chains of all the amino acids and was listed here. So as you can see, some of them have a high hydropathy, and they're usually the non-polar amino acids such as isoleucine, leucine, as well as valines and so on, as opposed to some like a polar amino acids such as acidic or the basic amino acids will have pretty low hydropathy, and they're going to have a polarity more on their side chains. So going through the protein residues from the first one in the N terminus to the last one in the C terminus, you can basically calculate the hydropathy based on the residues or the amino acid sequences, and can tell at some regions there's a high hydropathy or some low hydropathy. When there's a region there's a high hydropathy put together, that's more like the hydrophobic region that can be possibly buried inside of the protein, as opposed to some of the region that is more hydrophilic or with the low hydropathy values can be exposed outside of that globular protein, especially if that protein is located in the cytoplasm. To determine the 3D protein structure experimentally, X-ray crystallography is widely used. This X-ray crystallography is a technique in crystallography, but it uses X-ray too, to place the crystal that is formed in a specific condition. Sometimes it takes months to get this crystal too, depending on the molecular interactions of the protein or the substance that you're looking at. Anyway, in that case, there's an X-ray that lays on this crystal that goes through the lattice of atoms in this crystal. And then you can see the diffraction patterns of X-ray, which can be recorded and can be analyzed later on to predict the 3D structure of that. And this generally leads to an understanding of the material and molecular structure of a substance too. And nowadays, and there are different types of different resolutions that you can get depending on the stability of this molecules within the crystal. And the smaller the resolution, such as 1.1 angstrom resolution here, actually the better image you get, better prediction you get. And nowadays, there's a technique called cryo-EM. Cryo-electron microscopy is widely used to predict some macromolecular structures of not just focusing on one protein or the one substance. In that case, for the cryo-EM, since it has a slightly lower resolution than this X-ray crystallography, it'll visualize the bulky ones, such as nuclear envelope complexes. And another way to determine the protein structure is nuclear magnetic resonance, or NMR, which is a physical phenomenon based upon the quantum mechanical magnetic properties of an atom's nucleus. And NMR spectroscopy is one of the principal techniques used to obtain physical, chemical, electronic, and structural information about the molecules. And you can also analyze the dynamics of those molecules, as well as interactions between two molecules and their strength too. And it is a powerful technique that can provide detailed information on the topology, dynamics, and three-dimensional structure of the molecules in solution and in the solid state too. So it has a lot of powers. Of course, it has a limitation. It has to be limited in terms of the size of the molecules that it's looking at. It can be a little smaller than what you can get with the X-ray crystallography. gravity, and so on. But with the combination of all these techniques, you can get the pretty good idea and the information about the structure of the protein as well as their dynamics and interactions.